Dear students, myself, Dr. Supari Arham Javed, lecturer in Botany Department of Microbiology, BOTN 4128. Today, here is your lecture number 23, and we will start our new section, Control of Microorganisms, in which we will discuss today about principles and pattern of microbial death. First of all, we will discuss some contents in which we study some key points, then goal of microbial death, then what is microbial death, factors that affect the death rate of microorganisms, and then pattern of microbial death, rate of microbial death, and at last conclusion. We see some key points on which controlling microbial growth. Controlling microbial growth is important in the medical field and also in pharmaceutical and biotechnology industries, academic research and food industries also. And when we discuss microbial population death is exponential, and the effectiveness of an agent is not fixed but influenced by many environmental factors that surrounds us. Sterilization. Sterilization as a definition means that all the life was terminated, whereas sterilization and disinfection terminates selectively and partially. When we see chemical agents that can eliminate or suppress microbial life are separated in different groups based on their use. The major groups are disinfectants, antiseptics, and antibiotics. Then some antibacterials. Antibacterials are divided into two broad groups according to their biological effect on microorganisms. Bactericidal agents, or it may be bacteriostatic. Bactericidal, as the name indicates, they kill bacteria, while bacteriostatic agents, they just slow down or stall the bacterial growth or microbial growth. We see some review. In the previous chapters, we are concerned with microbial nutrition and growth, as you all know. But in this chapter, we address the subject of the control and destruction of the microorganisms, which is a topic of immense practical importance. Although most microorganisms are beneficial and necessary for human well-being, microbial activities may have undesirable consequences such as food spoilage and disease. Therefore, it is essential to be able to kill a wide variety of microorganisms. Our ability or inhibit their growth to minimize their destructive effects. The goal of microbial death is number 1, to destroy the pathogens and to prevent their transmission also. And the second one is to reduce or eliminate the microorganisms responsible for the contamination of water, food, and other substances. When we study about the microbial death, then we should have a concept of microbial death. That what is microbial death? It may be defined as the permanent loss of reproductive capabilities, or it may be defined as the cell cultures become dysfunctional, and it may be defined as the antimicrobial treatment leads to the killing of microbial population at the constant rate. Then factors that affect the death rate of microorganisms. It may include the time of exposure, that either we put lower temperature can be compensated with longer exposure. Second one, the number of microbes, that how many microbes will be present. Third one, microbial characteristics, that either it may be endospores or vegetative cells, or which agent is used. Last one is environmental influences, that either it is present in suspending medium, or what is the pH of the medium in which the microorganisms are present. Then, pattern of microbial death. A microbial population is not killed instantly when exposed to a lethal agent. Population death, like population growth, is generally exponential or logarithmic. That is, the population will be reduced by the same fraction at constant intervals. As you see in the table, a theoretical microbial heat killing experiment. If the logarithmic of the population number remaining is plotted against the time of exposure of the microorganism to the agent, then we will achieve a straight line plot, and when this population has been greatly reduced, the rate of killing may slow down due to the survival of a more resistant strain of the microorganism. Here you see a plot between the logarithmic number of survivors and minutes of exposure on x-axis. You see the minutes of exposure on x-axis and logarithm the number of survivors on y-axis. And when we put these on both the axes, we achieve a straight line. Here the d value. The d value is defined as how long it would take to reduce the bacterial population by 90% or 1 log at a given state of the killing agent. To study the effective lethal agent, one must be able to decide when microorganisms are dead, a task by no means as easy as with macroorganisms. It is hardly possible to take a bacterium's pulse, which is impossible. But a bacterium is often defined as dead if it does not grow and reproduce when inoculated into culture medium that would normally support its growth. In like manner, an inactive virus cannot infect a suitable host. As you all see, that this definition has flaws. However, it has been demonstrated that when bacteria are exposed to certain conditions that can remain alive but are temporarily unable to reproduce, when in this state they are referred as viable but not culturable condition. And this condition is seen in this figure, in this category. And this figure you see three categories, A, B and C. In the A category, cells leave stationary phase due to the starvation. And in B category, some believes that a fraction of a microbial population in this category. And B category, some believe that a fraction of a microbial population dies due to activation of programmed cell death genes. While in this C category, the viable but non-culturable category, The viable but non-culturable hypothesis posits that when cells are starved, they become temporarily non-culturable under laboratory conditions. When exposed, when exposed to appropriate conditions, some cells will regain the capacity to reproduce. While in the conventional tests to demonstrate killing by an antimicrobial agent, viable but non-culturable bacteria would be thought to be dead. This is a serious problem because after a period of recovery, the bacteria may regain their ability to reproduce and cause infection. Here you see the results of survivors in A category and survivors in B category, where the scientists thought that cells were killed after generating a programmed cell death. And here you see the survivors resuscitate upon change in the environment. Okay. When researching microbial death rate, the goal is usually to find out the minimum time needed to achieve acceptable level of sterilization for a specific purpose. And second, the D value, bacterial reduction, is determined by a D value, which is how long it would take to reproduce the bacterial population by 90% or 1 log at a given state of the killing agent. Microbial death curves have been developed for many agents and are used in numerous industries. Then, rate of microbial death. While we study rate of microbial death, the goal is to find out what is the minimum time needed to achieve acceptable level of sterilization for a specific purpose. The killing agent can be different, as for example heat. It may be chemical with certain concentration, and heat may be a moist heat or a dry heat. It's all depend upon the specific application. And second one, different killing factors, like when we see the heat, the phrase thermal death can be used. Thermal death or thermal death time is a concept used to determine how long it takes to kill a specific bacteria at a specific temperature. It was originally developed for food or food canning and has found applications in cosmetics and in producing salmonella-free feeds for animals. As for example, those animals which are present in our poultry and also responsible in our pharmaceuticals industry. Here you see a chart of thermal death time, which is plotted between number of cells and the time in seconds. The number of cells is present on the y-axis, while the time in seconds is present on the x-axis. And when we plot a table or graph, then we achieve a straight line, as you see in this figure. The data is obtained from the above table as is provided in your this lecture. When we see the thermal processing in food industry. In the food industry, it is very important to reduce the amount of microbes in products to ensure the proper food safety. This is usually done by thermal processing, which is not much expensive and is also very easy to apply, and finding ways to reduce the number of bacteria in the product. Time to temperature measurements of the bacterial reduction is determined by a D value, meaning how long it would take to reduce the bacterial population by 90% or 1 log at a given temperature, as I already told you about this. This D value reference point is 121 degrees centigrade. The D value reference is denoted by capital D with R. Here is our conclusion of our today's lecture, in which we study microbial death, factors affecting microbial death, pattern of microbial death, and D value. As a conclusion, we see that we can control our microorganisms by applying different factors like heat, which is also phrased as thermal death, and also by some environmental factors like pH, temperature, or some other environmental factors, which we will study in our next lectures, like some physical factors or some chemical factors. And we also see that when we plot a graph between number of exposure and between the number of survivors of our microorganisms, then we achieve a straight line. And we also see that microorganism may have a category of culturable, of viable but non-culturable category. This is all about your today's lecture. If you have any query about today's lecture, then you freely ask me in Google Classroom. Here are some references from where this lecture or your today's lecture is prepared. These all about from some your reference books like textbook of microbiology from essential microbiology, and also from principle and exploration of microbiology. If you want to obtain some other material, you may also contact me. Or if you have any query about today's lecture, then you freely ask me. Okay, dear class, thank you so much. Prepare your lecture. Best of luck.